This year, it's a pleasure to have Susanna Monge as our presenter, moderator for the scientific seminar. And over a year ago, when we decided to have vaccine and vaccination as the theme of our year, and then also of the theme of our seminar, we couldn't have thought of how timely and how important this topic would be over time. So they're not only disadvantages or suggested disadvantages, there are more advantages in vaccines. And we all know this, and vaccines are close to all of us who work in the field of public health. So I wish you a very good seminar, and hand over to Susanna Monge from the Instituto Carlos III in Spain. Susanna, please introduce the seminar.
Thank you, Ines, for the introduction and for the invitation to moderate this very exciting session, and thank you to all of you who are following this session either here on site or online. As introduced a bit by Dr. Stephens, we will be talking about vaccines. We will be talking about some exciting new developments with potential very high impact on public health. We will first go beyond what many of us, or how many of us, think about vaccines for infectious diseases, and go thinking about outside of the box on how this can be a very valuable tool for the prevention of antimicrobial resistance, which is one of the major current challenges we have in public health. And then we will be talking about also novel developments in the field of vaccines against vector-borne diseases, particularly chikungunya and dengue. So to take us through these topics, we are privileged to have three fantastic speakers. Two of them will be here on site with us, and one will connect online. And we also are very grateful for them, for the generosity to accept the invitation to speak here today. So without further ado, let me introduce the first speaker. He's Dr. Mateusz Hasso-Agopsowicz. He's working in the Department of Immunizations, Vaccines and Biologicals in the World Health Organization in Geneva. He's a vaccine immunologist, and we're very happy to be cross-cutting disciplines along this seminar. His work focuses on advancing the evidence-based policy, supporting development of new vaccines, and evaluating the role of immunizations on preventing antimicrobial resistance globally. So thank you, Mateusz, and the floor is yours.
Great. Thank you so much for this introduction. Good afternoon, everyone, and it's a great pleasure to welcome you here in my home country. I'm really happy that finally we have some infectious diseases conference that are happening in Poland, so welcome. I am Matis. I work for the World Health Organization. I work in the immunization department. And I think this session on vaccines is very, very timely, just because last year we have celebrated 50 years of establishing expanded program on immunization at WHO, 50 years ago. And in that time, we have estimated 154 million deaths have been prevented by using vaccines over 50 years. Huge impact. It's 6 deaths every minute for 50 years. So really, really huge impact. What I'm going to talk to you about today, that in addition to that really large estimated number, vaccines can do more than just averting deaths and averting diseases, and they can also impact antimicrobial resistance. I'll start with this first graph here, which is screenshot from a global burden of disease studies. It's a study done by IHME, Institute of Health Metrics, which looks at burden of various diseases, and they have also published estimates of burden of antimicrobial resistance. And in 2021, they have estimated that there are around 4.7 million deaths that are associated with AMR annually in 2021. And you can see on the right-hand side all the different pathogens that are contributing to this burden of antimicrobial resistance. On the very left, you can see that the 3 leading pathogens that are caused with associated AMR, they are causing around 2.3 million deaths. And these are Streptococcus pneumonia, Staph aureus, and E. coli. You can see this is a really large burden, and in order to tackle that AMR burden, we need many different interventions to appropriately do so. And one of these interventions could be vaccines. Vaccines could be used to reduce the burden of antimicrobial resistance. How do they do that? What are the different mechanisms for which we can use vaccines to reduce AMR? Firstly, vaccines can prevent both drug-susceptible but also drug-resistant infections. They can prevent you individually from becoming sick with a drug-resistant pathogens. But also, if sufficient number of people in communities are being vaccinated, they can also prevent those who are not vaccinated from becoming sick, just like the Streptococcus pneumonia vaccine, where we can see drop in disease incidence in those who are also not vaccinated. Because they reduce the disease incidence, they also reduce the antibiotic use, antibiotics that are often used to treat these diseases. And because of reduction in disease incidence and antibiotic use, they also reduce evolution of new drug-resistant genes, but also transmission of these genes to different bacteria and spread of antimicrobial resistance. And this is very nicely summarized in this graph here on the right-hand side, which is a screenshot of our action framework, which I'll talk about at the end of this presentation. When you think about vaccines and AMR, you may think, okay, I introduced my Streptococcus pneumonia vaccine to my country, I'll see high coverage, and eventually I will see some impact on incidence of drug-resistant isolates, or I may see impact on antibiotic use. But I wanted to show you on this graph that actually there are many, many more opportunities when one may think about what impact of vaccines on AMR may contribute in order to develop vaccines, to use them appropriately, to introduce them. And the boxes on the top are actually different stages of vaccine development, all the way from early development to developing a vaccine, then making a decision about a vaccine, whether to introduce it to a country, financing it, and then monitoring it after a vaccine introduction. And at the bottom, you have examples of various pathogens for which when development or licensure of vaccines was considered, for this particular pathogen, the role on AMR played an important, was an important factor. On the left, you have early development, and this is where for vaccines against, for example, gonorrhea or Klebsiella pneumonia, the impact on AMR was actually very important in making a decision whether to initiate work on these important vaccines. Then later, as you progress through clinical development, you have vaccines like Streptococcus pneumonia, where during clinical trials, very often the impact of vaccines on drug-resistant strains and isolates was being measured. And RSV and influenza, where during trials, very often impact on antibiotic use was being measured, and we saw that such vaccines have impact. Then as you move later into policy decision, for example, a vaccine against Salmonella typhi, the fact that it can be used to control outbreaks of drug-resistant Salmonella typhi in Pakistan, in Zimbabwe, was really an important factor to make a decision whether to introduce it in countries to help targeting some of these outbreaks. And then later, when it comes to Gavi, which is an international organization that purchases vaccines for low-income countries, in the decisions to decide as to which vaccines they purchase for low-income countries, the fact that vaccines can impact AMR makes a big decision on their evaluation framework. And then later, as you introduce vaccines, you can measure the impact on AMR after the introduction and also on antibiotic use. But despite understanding that the burden is really high because of AMR and showing you that at various stages of vaccine development, you can integrate AMR factors into your decision-making, we also looked at how vaccines are integrated into decision-making, to strategic plans to tackle AMR. And this is an analysis that was conducted by colleagues in the Netherlands, the NIVO Institute, which looked at something that is called National Action Plans on AMR. These are country strategies to explain which tools and interventions they're going to use in order to reduce AMR in their countries. And this research institute looked at 77 national action plans to measure how frequently vaccines are mentioned to reduce antimicrobial resistance. They found that, yes, a large proportion of them, they do mention vaccines, around 87% of them, but less than a half actually integrate them into national action plans, have indicators that would allow to measure how vaccines are effective in reducing AMR. I think what this means is that there is some interpretation, some understanding that vaccines do reduce AMR, but the integration of vaccines into AMR-related interventions and systems and strategies and plans is still lacking. And I believe that one of the gaps as to why this might have been caused is because for a very long time, we didn't really understand to what extent vaccines can reduce antimicrobial resistance. And this is what we had anticipated to do at the World Health Organization. We have modeled, together with a number of partners, what could be the potential impact of vaccines on reducing antimicrobial resistance. We looked at 24 different pathogens and various vaccines against these pathogens, and we found that the maximum potential of these evaluated vaccines to reduce AMR would be around 515,000 deaths, 28 million DALIs, $30 billion in hospital costs, $20 billion in productivity losses, all associated with AMR, could be averted annually if we were to widely use existing vaccines and develop new vaccines, in addition to averting around 2.5 billion doses of antibiotics every year. These are global estimates. And if you want to have a look further at these results, I invite you to look at the report that we had published. In the next 3 slides, I will go into a little bit more detail on these findings. The first one is there were findings in relation to how vaccines can reduce health-related AMR burden. And I've said that they could potentially avert up to 515,000 deaths associated with AMR. The largest proportion of these deaths is coming from the African region of the WHO. And on the right, you can see different pathogens for which vaccines are the largest contributors to these averted deaths. On the very left, you see tuberculosis vaccine, which is the biggest infectious disease killer, Klebsiella vaccine, and also E. coli, extra-intestinal pathogenic E. coli vaccine, followed by Streptococcus pneumonia vaccine. We have published these estimates in the BMG Global Health. I invite you to have a look and read in more detail. The second type of analysis that we did, we also look at what will be the potential impact of vaccines on averting antibiotic use. And we've estimated that it's around 2.5 billion doses annually that vaccines could avert. The largest proportion of that averted burden comes from the Southeast Asian region of the WHO, because you have very high number of people living there, but also very high antibiotic use in these populations. If you look at the graph on the right-hand side, you can see that the vaccine that contributes the most to that number is a TB vaccine. And this is because the TB treatment is incredibly long, often around 6 months at a time. You take plenty of antimicrobials, and because of that, that translates to very high overall use of antibiotics. And if we were to have a vaccine against that, then that could have really high impact. Secondly, we have extraintestinal pathogen E. coli vaccine, followed by Staph aureus, Group A Streptococcus, and also influenza vaccine. And the third type of analysis that we did is we wanted to understand hospital costs associated with AMR that are associated with treating the drug-resistant pathogen versus a drug-susceptible or versus having no infection. And what we have found is annually it is around $30 billion in hospital costs that are associated with treating drug-resistant infections annually, and the largest proportion of that came from the American region, unsurprisingly, given that in countries like the US and Canada, you have incredibly high costs of treating pathogens that are often require hospitalizations, long hospitalization, which is very, very costly. And that is also reflected on the picture on the right, which you can see that the largest contributor of a vaccine is a Staph aureus vaccine, which Staph aureus is often causing long, frequent hospitalizations, and that translates to very high costs of treatment. And this is why you can see a very high impact of such a vaccine on averting hospital costs. I've shown you just some examples of the results, but if you look at the report, there are many more ways of analyzing, interpreting results that you can look at for yourself. The first one is a dashboard for each of the vaccine that we have developed, which at the very top, it tells you what a vaccine is like, and then in the 3 tables just below it, it tells you what the vaccine-averted health burden is like, vaccine-averted antibiotic use, and then also vaccine-averted economic burden. And then we also developed dashboards online where you can look at one vaccine at a time and compare different impacts of that vaccine on AMR, but also compare multiple vaccines at the time and see, for your given interest, which vaccine would make the most or least impact. If you Google WHO vaccines and AMR, this should come up. What I have shown you so far is a lot of modeling analysis. So these are the modeling analysis that are based on AMR data, on vaccine assumptions, etc. But there are also a number of studies that have been published which show that vaccines can impact antimicrobial resistance. This is an observational study that is showing the impact of pneumococcal vaccines on the prevalence of non-susceptible invasive pneumococcal disease in USA. This is in children under 5. And what you can see here is that after the introduction of PCV13 vaccine, which is around 2010, after that, there was between 60 to 80% drop in resistant cases, resistant to macrolides, cephalosporins, tetracyclines, and penicillins. Now, this goes to 2013. If you were to observe that picture beyond and what is happening right now, you would see that these numbers, some of them would go up. This is because the disease, the pneumococcus that is causing the disease is the one that is not included in the vaccine. Therefore, you treat it with antibiotics, and often what you see is actually increase in resistance in some of these non-vaccine serotypes. But nevertheless, you can see the impact is quite striking. There was also another analysis that was done using data from demographic health surveys, which is surveys that is collecting health-related data in low and middle-income country settings. And they looked at what is the impact of pneumococcal conjugate vaccines and rotavirus vaccines on antibiotic use in children under 5 in low and middle-income countries. And this was published in Nature, and they had found that on the coverage during a time of this study, which I think was around 2020, so it should have been around 50% global coverage, they found that pneumococcal conjugate vaccine already prevents around 24 million antibiotic-treated episodes annually. And that if we were to increase the coverage of that vaccine to what WHO recommends, the 90% coverage in these settings, we could additionally avert up to 22 million doses of antibiotic-treated episodes. Similarly for rotavirus, with current coverage is around 14 million antibiotic-treated episodes. If we were to expand that coverage of what is required, what is recommended, 90% coverage, we could additionally prevent around 18 million antibiotic-treated episodes annually. I think what this shows is that we already have very good interventions that if we were to use them really well and extensively, these could have impact on antibiotic use and then later on antimicrobial resistance. Another data is from the RSV vaccine, and this was a Novavax vaccine trial which was conducted in 11 different countries. And this is a vaccine, it's a maternal vaccine, and what they have done is they've given the vaccine to the mother, and then they had looked at antibiotic use in neonates that were born to these mothers. And what they have seen is that there was reduced antimicrobial prescriptions for any diagnosis in infants who were born to mothers who received the RSV vaccine, around 13%. But if we were to zoom in on just prescriptions for acute lower respiratory infections, vaccine efficacy was around 17%. And you may think, well, that's not really a lot. But if you think of the overall number of antibiotic use, even small numbers, 10 and plus %, could actually have impact on the total antibiotic use that are being averted. And also in that time, during neonates and infants, this is the time where your microbiome is being developed. And taking antibiotics, it's really, you try to avoid it unnecessary taking of antibiotics because you wouldn't want to stop appropriate development of microbiome. This is within the first 3 months of infant's life. And another study, this one is coming from Canada. It was an ecological observational study where they had introduced free seasonal influenza vaccination to those 6 months and above in Ontario, Canada. But they didn't do that in other provinces. In other provinces, you had to pay for it. And what they found is that in the province in Ontario where they had introduced this vaccine, there was a 64% greater reduction in respiratory antibiotic prescription after that universal vaccine introduction, comparing to other provinces in Canada. And they've also seen reductions of secondary bacterial infections like pneumonia or otitis media. And you can see that on the graph. The bubble at the bottom on the right, this is the Ontario increasing with vaccination rates and also dropping in terms of antibiotic prescription. So what vaccines are actually being developed that we could use to reduce antimicrobial resistance? We did an analysis in 2022 where we looked at the vaccine pipeline to see for the WHO AMR priority pathogens, for which of them vaccines are being developed. And we found 61 different vaccines that were in active clinical development. Majority of them were for Streptococcus pneumonia, 16 vaccines. Not surprisingly, there is a high-income country market for these vaccines. There's a lot of interest, changing serotypes, etc. The second largest number of vaccine was for TB, 13 vaccines. There's a lot of donor support for this vaccine, recognition that this is the biggest infectious disease killer. And also 6 vaccines for Shigella and 6 vaccines for ETEC. But importantly, if you look right at the end, there are no vaccine candidates in development for some of these pathogens that are critical priority for AMR, so like Klebsiella pneumonia, Acinetobacter, Pseudomonas. And perhaps for some, vaccines may not be the answer, but it just shows you the real gap between what vaccines are being developed and what we currently have, and those that are critically important and for which we don't yet have vaccines in development. And that picture, since then, it has changed a little bit, but we're still in a dark period. Now, you think, when I talk to you about vaccines, how can we then think of vaccines and integrating them into global AMR strategies? And in WHO, we try to actively think how we can best recognize the impact of vaccines, how we can integrate them within the existing strategies, within the existing implementation plans. On the left, you have the Global Action Plan on AMR, which was published a long time ago in 2016. It does reflect the role of vaccines in terms of developing new innovations and vaccines, in terms of preventing infections and appropriately used vaccines. But now we are going through a time where we're actually revising the Global Action Plan on AMR. This is in response to the UN General Meeting that took place last year on AMR, which all the member states, they have signed a declaration to reduce the burden of AMR, and they have agreed on certain goals related to AMR. And in relation to these goals, we are now revising the Global Action Plan on AMR. What you see in the middle is a people-centered approach to address AMR in human health. And this is support to countries as to how they should be developing their national action plans to reduce AMR and what interventions they should be selecting, which ones are cost-effective in order to appropriately reduce AMR in their countries. And again, vaccines are prominently featured there. And last one is a global research agenda for AMR in human health. And this is identification of various research priorities, and again, vaccination is featuring there too. But in addition to that, we felt like perhaps this isn't sufficient. We need something else, something that is particularly speaking to the role of vaccines. And what we had developed together with experts is something that is called an action framework, which leverages vaccines to reduce antibiotic use and prevent AMR. This is a set of actions that we have published for policymakers, for pharmaceutical industry, for researchers, to think how in the daily work they could better integrate vaccines to reduce antimicrobial resistance. And these set of actions that are centered around 3 strategic goals. The first one is the need to expand the use of licensed vaccines to maximize impact on AMR. This is in relation to Salmonella typhi, pneumococcus vaccines, etc., and increasing their coverage. The second one is about developing new vaccines that contribute to prevention of AMR, like for TB, or think of vaccines that are very early in development, like for Klebsiella pneumonia, which could be very important for reducing AMR. And lastly, expanding and sharing knowledge of vaccine impact on AMR, recognizing there are still plenty data gaps, especially from low-income countries, which makes it really challenging for us to understand whether vaccines are appropriate interventions to reduce AMR. And that brings me to some conclusions. What we have shown in our modeling analysis is that vaccines have the potential to avert annually up to 515,000 deaths, $30 billion in hospital costs associated with AMR, in addition to $2.5 billion of antibiotic doses. I think importantly, the impact of vaccines on AMR, it needs to be appropriately recognized, both by AMR colleagues and stakeholders so that they can be integrated into AMR strategies, but also by the vaccine colleagues so that we can speak about vaccines not just averting deaths and infections, but also having much broader impact, including on antimicrobial resistance. And that the introduction of existing vaccines should be accelerated or expanded, such as for pneumococcus, such as for Salmonella typhi, and that we can prepare for introduction of newly developed vaccines, particularly for vaccines like for tuberculosis, which we're anticipating will have large impact on AMR, but that vaccine, which is primarily targeted at adults and adolescents to stop the development of TB disease to active form of disease, how are we going to introduce that vaccine? How are we going to appropriately use it? And also use that opportunity of introduction of such a vaccine to measure the impact on antimicrobial resistance. And lastly, think of some pathogens and vaccines that are very early in development. What is it for these pathogens that we would need to do in order to firstly understand whether vaccines for, let's say, Acinetobacter baumannii, are these the appropriate solution to AMR? What are the research priorities for some other vaccines, like for Klebsiella pneumonia? Or what such vaccines would need to look like? What would be the preferred product characteristics that would describe such a vaccine to make impact on antimicrobial resistance? So thinking really collectively about vaccines at various stages of development, starting from those that have been introduced, those that are in development, but also those that are very early in development. Thank you so much.
So thank you, Mateusz, for a very nice presentation. I think you showed very robust evidence of the impact that vaccines can have on antimicrobial resistance. So we have a few minutes for questions and discussion, and I would encourage people on site to do their questions with a microphone if we have one, and people from online can do the questions on the chat, but maybe while the microphone is sorted out and people type, maybe expand a bit, because I think it was very nicely visible in your presentation, the gap between the burden that some pathogens can have related to antimicrobial AMR and the pipeline of vaccines that are being developed. So is it any easy way to explain what are the... Is it any easy way to explain what are the barriers or challenges, or why is this development agenda being behind?
Yes, I think it very much depends on which vaccine you are looking at. You know, for some vaccines that are, for example, licensed, you have vaccine like for Salmonella typhi, which we know is very effective against drug-resistant infection. The challenge is that vaccine, there was a recommendation for this vaccine around 2019-18, just before COVID started. So as countries were preparing to introduce this vaccine, COVID happened, and that really delayed the considerations for introduction of this vaccine. Now we have times where we have really huge financial constraints in terms of vaccine introduction, and Gavi is saying that going forward we will need to prioritize which vaccines we can introduce to different countries. So again, we're facing another barrier where countries will need to decide, in fact, do I want a Salmonella typhi vaccine, or would I want to wait and for a GBS, group-based streptococcus vaccine, which may come in a few years' time, or a TB vaccine? So they need to start thinking in terms of prioritization. So finances, COVID and pandemic, that was one barrier. I think for other vaccines, earlier in development, like for TB, for example, group B streptococcus, it's also a matter whether you appropriately measure that data of vaccine impact on AMR during clinical development, if you have the opportunity to do so that during that development you can already collect that data, and then when it comes to making either a global decision or a country-level decision, you can already know, okay, I know TB vaccine demonstrated in trials, it impacts AMR. I have a big AMR burden for TB in my country, therefore I'll be more willing to accept and recommend this vaccine for use. And then you have vaccines which are very early in development, like for Klebsiella pneumonia, where you have plenty of challenges, different serotypes, K antigens, O antigens, they change in different regions of the country. You have hypervirulent Klebsiella, so many real biological challenges and technical challenges to developing vaccines, similar to Staph aureus, for example. So I think it really varies depending on which stage of vaccine pipeline you're looking at.
Okay, thanks. There's a question in the room? Please maybe present yourself briefly.
Yes. Vivian Bremer, Robert Koch Institute. Thank you for this really inspiring talk. I liked it very much. My question is about gonococci, because gonococci are one of the pathogens that are highly resistant and where we are at the end of a line with antibiotics. So I didn't see it really in your list. Therefore, I would like to know what were your, if you have thought about it, if it's appearing in your report, and if you see any chance.
Sure, it's a very good question. So yes, in fact, gonococcus, we recognize that it's a high priority for the World Health Organization. And in this report, we even have a separate note on gonococcus, because the data that we had used for this report, the underlying AMR burden data, was from the Global Burden of Disease study, which doesn't appropriately measure morbidity associated with gonococcus, such as loss of fertility, for example. And it doesn't report any mortality associated to gonococcus. And because of that, we felt like it doesn't appropriately reflect the burden of gonococcus. And if you then link it to various vaccination modeling, of course, then the vaccines would also not really show impact on AMR-related gonococcus. But there's been efforts that actually looked at, first of all, better understanding the burden of gonococcus in WHO, so incorporating all these additional morbidity outcomes that are not accounted for in the Global Burden of Disease study. But also, there have been a number of modeling studies which look specifically at gonococcus and model the impact of various vaccination scenarios if we were to give gonococcus vaccine in high-risk groups like MSM or women in low-income countries who are experiencing very high incidence of gonococcus, what impact could that have? And they found that because the transmission in these specific populations is incredibly high, that actually a vaccine with efficacy as low as 40% could actually have impact over a long period on antimicrobial resistance. So encouraging results. And given the fact that we have the meningitis B vaccine, which starts to show some protection against gonococcus, around 35 to 40%, and that could be a good one of the interventions that could be used.
Okay, so we have maybe time for taking one question online. So they say, thank you for your presentation. How can these findings be utilized to strengthen vaccine impact modeling and guide cost-effectiveness analysis for future vaccine investments?
I think one of the goals when we started this work wasn't just to come up with some modeling estimates, but actually to initiate conversations as to how we can integrate the impact of vaccines on AMR, all these different stages of the vaccine development that I have shown, and modeling impact of vaccines is one of them. Understanding the cost effectiveness is one of them. And I think we start seeing some results already. When we start talking about pathogens like group-based streptococcus, I see that actually discussions on how do we measure antibiotic use, that starts appearing. When we talk about vaccines against Shigella, which are in phase 2 development, the trial sites that are being prepared for a phase 3 trial, we start measuring antimicrobial resistant rates so that we can appropriately measure what should be the potential size of a trial to also capture impact on AMR, because we know that perhaps Shigella as a standalone vaccine may not be sufficient to make a decision about introduction of such a vaccine on its own. But if we add the impact on AMR, that may suddenly make a change. So I think we start seeing this increasingly, but I do recognize that what is really needed is some sort of a standardized way to come up with guidelines and metrics as to how we can integrate the impact on AMR into existing health economic assessments.
Yeah. So thank you. A lot of food for thought, I guess, for everyone in the room. Thanks a lot.
Thank you.
So we will move now to review some exciting advances in the field of vaccines against vector-borne diseases. And we will start with chikungunya. For that, we have Dr. Gerald McInerney from the Karolinska Institute in Sweden. He's a molecular biologist who has been working in alpha viruses, including chikungunya virus, for over 20 years. And the focus of his work is in how viral and host interactions at the molecular level affect the immune response and what are the implications this has for vaccine development. And today he's going to talk about how basic research informs the development of the chikungunya virus vaccine. So thank you, Gerald.
Thank you very much, and thank you to the organizers for this very kind invitation. So I'm going to talk about how basic research informed the development of the vaccine, or a chikungunya vaccine, for there are more than one. But so basic research. So I'm going to be showing some crystal structures of proteins, some fluorescence microscopy, and even a Western blot or two. So please, I know this is not what you can. I know this is not what most of you came to this conference for, but I hope in doing so, I hope to give you the impression that this was a curiosity-based project that really did lead to a clinical benefit. I also want to point out that I'm not the main character in this story. I'm here to represent the people who did do the work, but I'll explain my involvement as I go on. So first, with an introduction to the alpha viruses. So we're mainly going to be talking about chikungunya virus, but it is the main studied human pathogen within the alpha virus group. And this is a now globally distributed pathogen. So historically, the alpha viruses were quite well restricted to their regions. So the New World viruses were restricted to the Americas. For example, chikungunya was originally an African virus along with Onyongyong. And Ross River is over there in Asia, but chikungunya, as we all know, is now globally distributed and is now present wherever the Aedes mosquitoes are present. And this is just very quickly just to show the phylogeny of the viruses. So we do separate them into what we call the New World and the Old World viruses. And the Old World viruses are down here in this complex here, so including chikungunya, which is at the bottom here, and all of its other Old World viruses within this antigenic complex. So that's of relevance to the vaccine because this antibodies that react against these viruses tend to cross-react. It is one big serogroup. I won't go through this in too much detail, but just to show that the pathogenesis of chikungunya, of the alpha arthritic, primarily the Old World alpha viruses, but chikungunya in particular, initially delivered by an infected mosquito bite. And so you have a small amount of virus replication in cells in the skin, leading to an induction of an innate immune response and escape of the virus in a primary viremia, so it spreads throughout the body, essentially searching for tissues in which it can replicate more efficiently. And it does so, this virus does so in the joints. So in certain cell types in the joints and in the bones, this virus replicates very well. And then you have a secondary viremia through which the virus can then be picked up by a second mosquito and then thus completing the life cycle. And so this tropism of the virus to the joints and to the joint tissues is of some relevance for the pathogenesis of the disease. So the disease in the acute phase is very similar to a lot of other tropical diseases, and historically, chikungunya, dengue, and Zika have been treated very similarly, and maybe we think there's quite a lot of underreporting of chikungunya because of that. But it's febrile illness is characterized by a rash and a certain amount of malaise and general symptoms of tropical diseases. But what separates chikungunya from these other diseases is this joint pain, and this is, of course, a consequence of this replication in the joints. And so a rather large number of people who've experienced the acute phase will then experience what we call the chronic phase, where you have replication, pain in the joints, fingers, hands, knees, and all of the joints in the limbs, and which gives chikungunya its name. In the Makonde language of Tanzania, chikungunya, I believe, means to be contorted. And so it describes the posture of the people who would have that disease because their joints are so painful. And historically, we haven't really thought of chikungunya as being a disease that has a significant mortality. But in an outbreak in Mauritius in 2006, it was, I don't want to go through this in too much detail, but just show that there was a significant increase in the observed mortality on the island, which correlated very well with the peak of the incidence of chikungunya, as you can see down there. So we do now believe that there is a significant mortality with chikungunya that goes along with these other symptoms. Okay, so the natural history of the virus, as I said, it emerged or was first discovered in Central and Eastern Africa, and then soon after a similar virus was detected in Western Africa, chikungunya, Western African isolate, and then through global travel in the 1950s and 60s, the virus spread, transmitted by Aedes aegypti mosquitoes through Asia. And so following the red circles there in Asia. And then sometime in the early 2000s, 2005-06, the virus picked up a single point mutation in the spike protein, which allowed this virus now to enter and replicate in cells of the Aedes albopictus, the tiger mosquito, which is much more aggressive mosquito, which now allowed the emergence of new strains of the virus in Asia, and then very soon after that, the emergence of the virus in the Americas, and where hundreds of millions of people were infected. And the virus is now globally distributed, providing to us an example of any of these viruses, which I mentioned are generally restricted to certain regions, but with the potential to emerge and to go global. And just the last slide on the transmission, these are the Aedes albopictus and aegypti, and as of June 2025, according to the ECDC website, the Aedes albopictus mosquito is very comfortably moved into Europe and expanding. So you see all the red there is regions of established populations of Aedes albopictus, and it's no surprise then that there was a chikungunya outbreak earlier this year in southern France, and there will be more as the years go on. So moving to the molecular biology, so this is just a scheme of the intracellular life cycle. So this is a virus that enters by receptor-mediated endocytosis, and it's a positive strand RNA virus, which is of relevance for something I'm going to mention later, which means that the genome of the virus is also an mRNA, and it's translated directly. And the first proteins that are made are depicted over here. And these are the so-called non-structural proteins, the proteins that form the viral polymerase complex, and also form, that communicate with the cell, that bind to host factors and communicate with the cell, reprogramming the cell in order to become a factory for making virus particles. These are very, very fast replicating viruses. And once all that is done, then what's happening in the center here is that the RNA replication becomes very, very efficient, and the cell just floods, the cytosol is flooded with viral RNA, and the viral protein production is very, very efficient, and many copies, hundreds of thousands of copies of mature virions are released per cell. So there's a vast amount of virus replication going on. In recent years, there's been a lot of work on the structure and molecular functions of these viral replication complexes. This is the NSPs 1, 2, 3, and 4. Don't need to follow this in too much detail, but just to say that we now know at pretty much atomic level the positions of these proteins and how they form these membranous buds on the plasma membrane where viral replication occurs inside these buds that bulge out into the extracellular environment. And we know the positions of these proteins, NSP1, 2, and 4 particularly, and NSP3 is a little bit less well understood. Just to go on a little bit, so these replication complexes that form on the plasma membrane, in some of the viruses, not all, but in some of these viruses, they're then internalized, so you get these vesicles that form in the cells that look like the electron micrograph there, which are just machines for making viral RNA. Can you imagine this in 3 dimensions, with each one of those little bulbs with the black dot is producing RNA, and the cell is just flooding with viral RNA, which is the reason why the viruses replicate so fast. And just of interest at this point, I want to point out that this internalization of these replication complexes, we can detect it with an antibody to double-stranded RNA, and it's dependent on the cellular AKT pathway, which is a cellular metabolic pathway. And the reason why I'm mentioning that will become clear in a minute. So the NSP3 protein, historically referred to as the enigmatic NSP, non-structural protein, we didn't really understood what it's doing. It has 3, sorry, I keep pressing the... It has 3 domains, the 2 on the left you can ignore, but the C-terminal domain is referred to as a hypervariable domain, and this is a sequence that's different in every alpha virus. It's really completely unconserved, except for some short linear motifs, which I've shown there. There's YXXM in particular I'm going to mention. And these are the motifs that bind to cellular proteins and help the virus to communicate with the cell and to turn off cellular proteins, right, cellular pathways. So we had been working on NSP3 in alpha viruses generally, but a project I want to tell you about was done in this other virus called Semliki forest virus, where there was a virus called Delta 50, which has this large deletion in NSP3. And we were studying this in terms of this AKT pathway, and we found that the Delta 50 virus behaves like a wild-type virus under AKT inhibition. So suggesting that something in that deleted region is activating the AKT pathway. And cutting a very, very long story short, we found that it was this tyrosine that we mutated to phenylalanine in this mutant. And here's the one Western blot that I promised. And so you can see that on the left, wild-type virus activates AKT very, very strongly at just 4 hours. The first bar is 4 hours after infection. This is a metabolic pathway is turned on. The cells are just producing protein. But the mutant fails to do that, and the mutant grows to much less titer, although it does replicate. And so that allowed us to propose the following, and a lot of work that came downstream, showing that through molecular mimicry, this NSP3 protein can bypass the need for growth factor receptors and push the cells into a so-called pro-survival metabolic pathway. So this is cell division, lots of protein synthesis, and this is a very well-studied pathway in cancer development because pushing cells into cell division is very bad for causes tumorigenesis. But here we're showing what the virus is doing. So in parallel to this, my colleagues Peter Lilleström and at Karolinska and Andres Merits in Estonia decided that they wanted to make a chikungunya virus that looked similar to this Delta 50. So in other words, recapitulating this mutation in the NSP3, and they did that. And what they found was a virus which they called Delta 5, which just like our Delta 50 mutant, it does replicate very well, although the plaque sizes are slightly smaller, which means that the virus is not replicating as well. In mice, the viremia was lower in most of the animals, but in some, there was some sort of intermediate. But the foot swelling, which is a symptom of the infection in mice, was much less in the virus, in the mutant. And then typically, or importantly, if these mice were immunized first with Delta 5 and then challenged with wild-type virus, there was no detectable viremia at all. That's down here. So no detectable viremia if the mice first experienced this Delta 5 mutation. And the same in the macaques. If they gave macaques just PBS and then challenged them with wild-type chick, they had a very strong viremia. But if previously immunized with Delta 5, no viremia was detected. So this was a pivotal result that allowed that made it obvious that this was a very good candidate for a vaccine. And that's exactly what Valneva went ahead and did. They copied the Delta 5 mutation, but now it's called VLA1553. And it was developed as a live attenuated virus particle vaccine. In other words, it's delivered as virus particles. So I'm not connected with Valneva in any way, so I don't want to talk too much about this, but there's a very nice article where you can read about the phase 1 study and the phase 3, both of which showed very strong seroconversion and very strong antibody responses, and leading to US approval and also approval in Europe. I can also point out that very recently in September, Valneva reported that even 4 years after a single shot of this vaccine, still 95% of the vaccinees have antibody responses. So this is very optimistic, although there has been some optimism has been dampened by some adverse effects that were detected in some elderly patients and some vaccinees in some of the trials. So some caution should be used when deciding which groups should be vaccinated with this vaccine. Nevertheless, we were following this at Karolinska with great interest because of the history of the development. And then when it became available in Europe, my research group and I, there's the 5 of us down here, we all went to get the vaccine. We were working with the virus in our lab, so we wanted to take the vaccine primarily as to protect ourselves from the virus, but also we reasoned that this was a nice way to celebrate basic research. And even though none of us in that picture were involved in that particular virus, we were there to represent them. And we brought with us a journalist from our institute and a photographer, and they made this nice, and I told the journalist many times, I'm not the guy who made this vaccine, but nevertheless, the headline was, researchers take their own vaccine, which is, so again, caveat, I'm here to collect all the credit, but I didn't do the work. And then I'll leave you with this last slide that Peter and his group also developed. So the Valneva vaccine is delivered as a particle vaccine. So this vaccine has to be propagated in industrial tissue culture conditions, which is problematic in itself, but also the potential that it may lead to reversions or other mutations. But what Peter's group also showed is that now you can deliver this vaccine as RNA, because this is a positive strand RNA virus. So just injecting the RNA or delivering the RNA into cells should initiate a virus infection. And that's exactly what they did. So they made these constructs, which is called iRNA or iDNA. And so you can introduce into animals, and this is in mice, without any adjuvant or without any nanoparticles, just the naked RNA into the muscle, so 1 microgram. And they found that, so this is the same mutation, this Delta 5, and you get a very strong antibody response, less so with the DNA. The DNA is in blue here, so less of an antibody response with the DNA. Those antibodies are neutralizing, and then 9 out of 10 of the mice that receive the RNA were protected from chikungunya challenge. So I thought this was an interesting thing to flag up. These kinds of approaches could be used for delivering RNA. Now that there's a global network of infrastructure for delivering mRNA vaccines, we can now also, its potential to deliver live attenuated virus vaccines using that technology. So thank you all for your attention, and my main thanks should go to my colleagues Peter Lilleström, Andres Meritz, and to a lesser extent Roger Legrand, who were the developers of the Chick Delta 5 vaccine, and David Hallenjard and Inge Surzot and Kalle Ljungberg were the lab guys. This is my group down here, and we continue to work on NSP3, trying to figure out what that deletion, that region is doing, and other regions. And these are our funding agencies on the right. And I just want to point out that we do have a PhD student position available. If anyone is interested or knows anyone who's interested, please let us know. Yeah, thank you very much for your attention.
So, thank you very much for this impressive work and presentation. Do we have any question in the room? No? Yeah. Yes, it's coming to you.
Thank you for your nice presentation. I'm Daniel from Maastricht University. You were explaining the rapid development of chikungunya and the different serotypes. But Jan Semenza also explained the rapid uprise of mosquitoes in especially Europe. Do you expect us as public health to have a seasonal vaccination campaign in the near future? Or what are your thoughts on the vaccination and public health vaccination programs?
That's very far from my expertise, so I really don't think I should comment on that. I mean, it's certainly true that we'll have more outbreaks of this virus in Europe. I think my gut feeling is that the vaccine should be taken in the event of an outbreak rather than seasonal. Yeah, but again, I'm sure half of the room could comment better than I could.
So maybe ask, because you presented some serological data, neutralization assays, etc., but is always the question about what are the correlates for protection and what's the clinical significance of the antibodies that we can detect with these assays. So is there clinical results on the effectiveness of this vaccine?
So there haven't been efficacy studies done. So the vaccines were approved, to my knowledge, based on the strong antibody response and perhaps the assumption that the correlate of protection is neutralizing antibodies, but it's of course more complicated than that. But the antibody response is very strong, and is, at least in all of the animal studies, you saw the viremia is really undetectable. Mm-hmm. So yeah, I think part of the hesitancy with this vaccine, and part of the reason why it maybe hasn't been approved, it has been withdrawn in some regions, is because of the lack of such studies. Mm-hmm. Yeah.
So maybe going back to the molecular aspect of it, and I'm an epidemiologist with limited understanding. So for people like me, is it any easy way to understand what this fragment that you took out from the virus is doing? Why is this attenuated? And then, if I understood correctly, this is this NSP3 is a protein that's shared between many alpha viruses. So does it mean that this can potentially be expanded to other viruses in this family?
It's a very good question. The region that's deleted, I mean, I put it in the context of what we know about the other virus, Semliki forest virus, but actually we don't know exactly what that sequence is doing. Mm-hmm. And the sequence in the other viruses, Onyongyong and Ross River, are different sequences, but there's probably functional overlaps. And I would be quite confident that deleting the regions in the other alpha viruses would pretty high degree of confidence would lead to a virus of similar phenotype. Whether it would be a better vaccine or not is hard to tell. But I think that the approach of using infectious RNA is something that is by making targeted deletions in regions of the virus that are communicating with the cell. So reprogramming the cell. If you can stop the virus from reprogramming the cell, you make a virus with a different phenotype that might be more immunogenic, which this one is. And so for flaviviruses or even coronaviruses, there are now infectious RNA systems that could be harnessed for this kind of analogous approaches. So it's not something we're working on, but I suspect that others are, and I look forward to seeing that more of that in the future.
Okay. So maybe take one last question from the online questions. They thank you for the presentation. And this goes a bit in the line of what we were discussing now. So it's clarification about immunogenicity, efficacy, safety, costs, and benefits. So I guess the question is like a more overall assessment of this vaccine from even the implementation point of view, not just immunogenicity and efficacy that we talked about, but also safety and cost and benefits, which is something that when you arrive to implementation in a public health program would consider.
Yeah, I mean, it's a live attenuated vaccine, so there will be a different set of safety issues with this compared to subunit vaccines or protein vaccines. So I think that has to be taken into account, and it's clear that there has been some adverse effects. So a certain amount of caution is warranted, as with all live attenuated vaccines, I think. And from the basic research to the approval of the vaccine was very, very quick, and indeed there's more studies that need to be done to secure the safety and to secure the decision making going forward in certain regions.
Okay, thank you very much. Then thank you.
Thank you very much.
So we may go to our third and last speaker for this session. She's online, so I hope we can see. So our third speaker is Dr. Fernanda Boldos. She's Chief Medical Officer at the Butantan Institute in São Paulo, Brazil. And she's been leading clinical programs for evaluation of the Butantan dengue vaccine, which is currently under regulatory approval for medicines agencies, and also for the chikungunya virus vaccine. But today she's going to talk about dengue strategies for prevention and control. So welcome, Fernanda, and over to you.
Thank you very much. I hope you can hear me well. Please let me know otherwise. So it's a pleasure being here. I'm sorry that I couldn't be there in person, but hope that we can have a nice discussion around dengue and a new vaccine from Instituto Butantan. So today we are going to be talking a little bit about dengue epidemiology and why dengue is becoming a global disease, much more than an endemic country only problem. So when we can see this figure that I'm showing this slide, we can see that we have at this point more than 100 countries where dengue is endemic, that poses 400 million infections annually. Only 1/4 of the cases are symptomatic with dengue. We have around 75% of the infections being asymptomatic, but it's still leading to more infections, of course, that really increases the force of infection of the disease. And we have around 40,000 deaths annually caused by dengue. That really poses a burden that is social, economic, and we have at this point 50% of the population in the globe that are susceptible to having exposure to dengue. So I really believe that this is becoming more and more a global threat. This is also, one of the reasons is the climate changes that we have been seeing, but I'm going to briefly speak about others. So we have been seeing here that countries that initially were not in the list of the countries to have autochthonal cases of dengue. Now we have been seeing more and more cases, autochthonal cases in the global, in the North Global of. And this really imposes a problem. So we do have 60.1 billion that will be at risk of having exposure to dengue by 2080. And this is because diverse factors, as I mentioned, we have urbanization, we have globalization and travels, and that really brings different serotypes of dengue for susceptible populations. We have the climate changes, as I just mentioned, so we really have warmer temperatures that really accelerate mosquito development, increase viral replication, while we also have the extended rainy seasons that are ideal conditions for the mosquitoes to replicate. And also we have difficult on controlling the vectors. So the measures that we have nowadays to control the mosquitoes are not effective. So here is just to quickly show that with the global warming, we are seeing the altitudinal range that is changing. And with that, mosquitoes are going to be able to be in highest places where they couldn't replicate before. So that would be leading to cases in places and countries where dengue was not seen before. When we look to an endemic country as Brazil, where we have annual seasons of dengue that occur during the South summer, so between November and March, that's our peak for dengue, we also have seen a different pattern. So when we see those light blues, 2023 and 2025, that is kind of our seasonal peaks of dengue that we are usually to deal with. But in 2024, last year, we had an unprecedented outbreak that led to 6.6 million suspected cases of dengue. All of them, we are talking about febrile cases in here, and more than 6,000 deaths confirmed due to dengue in 2024. So this pattern really led to a breakdown on the hospital system, on the healthcare system. So that's something that we are also looking into this huge impact in countries that even in endemic countries that are used to deal with dengue. So talking a little bit about the disease per se, so that I can share also in the prevention measures that we have afterwards. So the dengue virus is a flavivirus, and dengue has 4 distinct serotypes that we call them 1, 2, 3, and 4. And the presence of 4 distinct serotypes really leads to a lot of challenges in terms of vaccine development that I will detail in a few slides. The primary vector for dengue transmission is the Aedes aegypti mosquito that is very adapted to urban areas and bites primarily during the day. But we also have a secondary vector, which is Aedes albopictus. And this vector is responsible for the number of cases increasing in the north region because it can adapt to more temperate regions. So with the changes in the altitudinal range and the temperatures, Aedes albopictus is expanding more and more to the north side of the global, as you can see in the figure. When we look to the clinical spectrum of the disease, as I mentioned before, 75% of the cases are asymptomatic. which of course leads to a lot of under notification, and that impacts surveillance per se. And the 25% of the cases that become symptomatic, the majority of them are a dengue fever, that it poses with a sudden high fever, headache, pain behind the eyes, muscle pain, rash, that is very common, and nausea. But what can happen with dengue, it is a severe cases or dengue hemorrhagic fever, as it was called before, when we have after this febrile stage, that during the first 3 or 4 days after infection, we can see a plasma leakage that leads to fluid accumulation and can cause impairment in diverse systems and organs that can be fatal as well. That happens when we have already the viremia going down, and can be very critical. The majority of the deaths from dengue are due to missed warning signs and late presentation of symptoms. At this point, we do not have any specific treatments for dengue, so the treatment is symptomatic only. And this gap really causes a need for more effective prevention tools. The severe disease can be also related to a condition that we call antibody-dependent enhancement. And that is related to the fact that I mentioned before that the dengue virus has 4 different serotypes. So when someone is infected by one serotype, this natural infection really can provide lifelong immunity to that specific serotype, but not the other ones. So when this person is infected by a second serotype, the organism can recognize this second type of virus, but cannot neutralize it appropriately. So that leads to increased viral load that can lead to severe disease. That's what we call antibody-dependent enhancement. And that the main reason on why a dengue vaccine, a development of dengue vaccine poses so many challenges, because we need to have a balanced vaccine against the 4 serotypes. If the vaccine is not balanced against the 4 serotypes, we can really facilitate the ADE, the antibody-dependent enhancement, because with the vaccine, we are providing the immunity against 1 or 2 serotypes, and then when the person is infected by a different serotype, can have a severe disease, because the vaccine can act as this primary infection. So we really need a vaccine that is balanced against the 4 serotypes to ensure that we are not causing ADE as it happened with other dengue vaccines before. So when we look to the pillars of dengue prevention and control, we have 4 pillars. The first one is surveillance and outbreak preparedness. Then we have the vector control, vaccine and therapeutics. We do have some vaccines already commercialized, but therapeutics, all of them in development stage, specific therapeutics, and community engagement. Talking briefly about the surveillance, we really need to move for more of an active surveillance than a passive one. So we can really try to combine all the to generate epidemiological data that we will need, really to understand not only the number of cases, but also the serotypes that are circulating in each one of the specific regions. Then we could integrate that data, this epidemiological data, with entomological data. So we need to understand the mosquito indices, the rain seasons, and etc. So we can try to predict where the outbreaks will be happening, so we can try to act early on. So that would be the ideal. In terms of diagnostics, we need to have also rapid diagnostic tests for screening, and then PCR for confirmation and serotyping. And of course that we need to have all of them, all these surveillance measures in place, so we can really have a data-driven decision in terms of where we are going to pose our efforts to avoid huge outbreaks caused by dengue. And in terms of vector control, besides what we have already been familiar with, that is not allowing a lot of water to be stagnant water that we know that is very good for mosquitoes and insecticides and etc. We do have a new measure that is called Wolbachia, which has been showing to be, it is now being implemented. We are now having the studies to understand the real impact that this is having, but this is kind of hypothetically can be an important auxiliary method to decrease the amount of mosquitoes that can cause dengue in a specific region. So, in reducing the ability, because this is kind of infecting the male Aedes with Wolbachia bacterium. When they mate with a female, they produce eggs that cannot hatch. So with that, we can reduce the ability of those mosquitoes to transmit the virus. So these male Aedes mosquitoes that are released in the environment can really avoid that. We can keep increasing the ability of the mosquitoes to transmit the virus. Nice things about this is that there is no environment impact. There is not NGO, and there is no safety risks related to that measure. So studies have shown, initial studies, pilot studies have shown a decrease on the number of dengue cases in the region where those mosquitoes were released in the magnitude of 70%. And now we are having more studies evaluating these kind of strategies for us to ensure that these can be effective as a complementary strategy to decrease the number of cases. And now going to another preventative tool, which are the vaccines that we have been discussing. I'm going to show the data of Butantan DV. That's the dengue vaccine that we have been developing for the last 15 years at Butantan. And as Susanna mentioned in the presentation, we ended phase 3, and we are now waiting for regulatory approval in Brazil, which would be the first country where this vaccine could be potentially approved. So this is a technology that was licensed by NIH, is based on TV003 from NIH, and NIH licensed that for different companies, as you can see in the figure, Instituto Butantan being one of them, and we have the rights of the use of this in Brazil and other companies in different parts of the world. So Butantan DV is a live attenuated tetravalent dengue vaccine candidate that is analogous to TV003 that was developed by US NIH in the lab of Steve Whitehead. And as you can see in the figure, we have the full virus strain for DEN 1, DEN 3, and DEN 4. And DEN 2 vaccine virus strain is a chimeric virus, which is based on DEN 4 backbone, but encodes the DEN 2 structural proteins. So all of them are attenuated by having the delta 30 in the 3 ends. And as I mentioned, as you can see in the figure, DEN 2 is the only one that is the chimeric one. In terms of clinical development, so after license these from NIH, and I will go quickly from the phase 2 and initial phase 3 results, because that was released before in different meetings. I hope that, and it is published, and I will share the publications in here if someone wants to go deeply on that. But I will also show the 5-year results that is kind of fresh news. We released them for the very first time last week in Toronto at the TropMed Congress, and that's the second time that we'll be showing that data. So phase 2 really confirmed that the vaccine was well tolerated and immunogenic in a phase 2 that we did in 2014. Then in 2016, with those results, we started a phase 3 that was an efficacy trial based on clinical outcomes, where we were evaluated a single dose, that is a single dose vaccine of Butantan DV or placebo in a randomization 2-to-1, where we had enrolled participants from 2 to 59 years old and followed them up for 5 years to ensure that we were not seeing any signs of ADE. That's the reason on this long follow-up. And that was, of course, aligned with the regulatory agencies. In terms of participant disposition, we were able to enroll 16,235 participants in the age groups that you are seeing here, so very well balanced in terms of the age groups. The study was conducted by 16 clinical sites across Brazil, all of them in the public healthcare system. And the randomization rates are, as you can see in here, so 10,000 plus participants received the vaccine. So we do have different cutoffs in here that I'm going to show briefly. So the first one is the 2-year. That was the primary endpoint of the study. So efficacy against any virological confirmed dengue. So we are not talking about severe dengue only. We are talking about symptomatic diseases after 2 years of a single shot. Then we have another cutoff that is this lighter orange in there. That is the same date cutoff, but a lot of those participants had already completed more than the 2 years because the randomization had a de-escalating approach to include the kids, mainly the smaller kids. So the average of the second follow-up, it was 3.7 years, and then I will end with the fresh 5-year data as well. So in terms of the overall efficacy after 2 years primary endpoint, we have shown efficacy against virological confirmed dengue cases of 79.6%. This was published February 2024 at the New England Journal of Medicine, so you can have more details on that. And just to mention this figure quickly, because this was in the 3.7-year paper that is, I will show in the next slide that was published in the Lancet. But I think that was an important feedback that we received after the first paper on how we should also shun the data by calendar time, not only after the vaccination. So that's why we landed on putting this graph. So quickly going through this, each open circle that you can see represents a VCD case, a virologic confirmed dengue case. So we have a total of 356 cases in the study at this cutoff data, 3.7 years. The colors are for differentiated regions where sites were from, because of course, although dengue is endemic in the entire country, do have different behaviors in each one of the seasons, and the regions can really define where the cases are happening. So we have seen that. So we haven't had any cases in the south of Brazil, for example. The x-axis represents actual calendar date of a dengue cases, while the y-axis is the onset of a dengue cases since vaccination, measured in days. And the leftmost vertical dashed line is when the enrollment began, and the middle vertical dashed line is when the last participant was randomized for these 2 and 3.7 years cutoff. Okay, so with that we can see how the cases were distributed throughout the studies, looking both to days after the vaccine and also calendar days. Then here it is the 3.7 years follow-up data. So we have seen the overall efficacy against virologic confirmed dengue after 3.7 years of follow-up was 67.3%, keeping the same safety profile. And this data was published in the Lancet ID. So the paper is also here if someone wants to have a deeper look onto that. So here I'm showing combined data from 2 years and 3.7 years. So when we look to the overall efficacy, as I mentioned, 79.6 and 67.3 in the two cases, but here we're also looking at data per serotype. So we, for DEN 1 and DEN 2, as you can see in the figure, first table 2 years, second one 3.7 years. And those ones that are in the red box, they are related to the primary endpoint, and those are the endpoints that had in the trial a pre-specified hypothesis testing, that the lower bound of the CI needed to be higher than 25%. So as you can see, that was achieved. So the study met the primary endpoints on showing the efficacy against virologic confirmed dengue. And a limitation that we had in this study is that there were no cases of dengue 3 and dengue 4 during the follow-up of the study, which was the epidemiological situation in Brazil in the years while the study was there. We didn't have big outbreaks on dengue 3 and dengue 4, so unfortunately we had this limitation. So here, when we look to the efficacy by age group, we can see that at 2 years, we have here adult, then the 7 to 17 years old, and then the smaller kids, 2 to 6 years old, and then the efficacy at 2 years and the efficacy at 3.7 years. You can see that in general, the efficacy is maintained. As expected, we do have folks with prior exposure to dengue having an efficacy and a higher efficacy than folks that haven't never been exposed to dengue. And one of the beauties of this study that we consider that this was a very stringent test for the vaccine is the fact that we have 46% of the study population that was dengue naive, so never been exposed to dengue before. And here we have the 5 years follow-up data. As I mentioned, this is fresh data that was released last week for the very first time, and the paper is still embargoed, but should be out in the next weeks. So we have an efficacy, overall efficacy after 5 years of follow-up of a single shot of the Butantan DV, that is 65% of protection, being 77 when we are looking to this population with prior exposure, and 58.9 in the population without prior exposure to dengue. And we look to dengue 1 and dengue 2. This is the data as well. Both in the 2 years, 3.7, and now in the 5 years, we do see a dengue 1 being more effective than dengue 2. And if you just remind of my slide on the structure of the vaccine, dengue 2 was the only chimeric protein. Dengue 1, 3, and 4 are the full virus strains. So that was something that in terms of biological plausibility, we would expect that dengue 2 would be maybe our I would say less effective serotype, but even though we do have interesting results even after 5 years of follow-up. Yeah, just to show here again what it was, the pre-specified hypothesis for the 5 year. And I think here this is one of the most important data that we have been shown. So after 5 years follow-up, we had 80.5% of efficacy on preventing severe dengue or dengue with warning cases, and 100% of efficacy on preventing hospitalization due to dengue. So some of the dengue with warning sign cases, they don't need to be hospitalized. So that's the gap between 80.5 and 100% results. In terms of safety, we have been seeing the solicited systemic vaccine-related AEs were observed in a slightly higher proportion of participants receiving the vaccine. 58.3 compared to 45.6 in the placebo group within the first 21 days post-vaccination. During the follow-up, we had 67 deaths on total, but none of them were related to study treatment. And we did conduct the study during the pandemic, so a majority of those cases were related to COVID. And the systemic is were the only one that we had a more important imbalance, and I will show in the next slide which were those. So what we have been seeing in terms of unbalance between placebo and vaccine in terms of systemic AEs were mainly rash and pruritus. So as, of course, as dengue, the vaccine, because it is a live attenuated vaccine virus, we do have also these rash and pruritus, but biggest majority of them were mild and self-limited with a duration, median average duration of 3 days. So in summary, in terms of the vaccine data, we have shown that a single dose of Butantan DDV was efficacious against symptomatic virologic confirmed dengue due to DEN 1 and DEN 2, regardless of baseline serostatus or age, throughout 5 years of follow-up. As I mentioned, we had a limitation of not having cases of 3 and 4 during the entire study period, and the safety profile was consistent with previous study, very well tolerated. No safety concerns were observed during the entire 5 years follow-up. Important information was that there was no imbalance in the proportion of participants with dengue with warning signs or severe dengue between intervention groups. And Butantan DV demonstrated efficacy against dengue with warning signs and severe dengue, and against dengue hospitalization due to dengue. The study, as I mentioned before, covered a wide range of 2 to 59 years of age and included a substantial proportion of participants without evidence of prior exposure to dengue. So that really put us in a situation where we think that this can be an important tool in terms of dengue prevention. So I see that my time is due, but I'm also finished. So thank you very much. I'm available for any questions that you may have.
So thank you so much, Fernanda, for this comprehensive review, and particularly for the fresh results in the 5-year follow-up. I think we're privileged to be seeing these results already. So we are out of time, but maybe one question for Fernanda from the room. Otherwise, maybe Sabrina.
Thank you, Fernanda. Sabrina Bacci from ECDC. How would you see this vaccine incorporated in the childhood program? Is it a crowded program in Brazil? Do you have any considerations around this? Thanks.
So how this vaccine would be implemented in the immunization program? That was the question?
Yes.
Yes, okay. So yeah, that's definitely a need. So although we do have an approved vaccine for dengue, we have 200 million people in Brazil, all of them living at risk of dengue. And what we had with the approved commercial vaccine that is already incorporated in the immunization program in Brazil, that we just had 5 million of doses, and that is a 2-dose vaccine. So we're able to vaccinate 2.5 million of people out of 200 million. So non-impact in terms of epidemiological impact in terms of disease transmission. So we are a public lab. So everything that we do kind of in partnership with the MOH. So we do know that this is an important need, and MOH is willing to incorporate that vaccine in the national immunization program in Brazil as soon as we have the regulatory approval that should happen in the next weeks. So we do see that this will be a massive use. Of course, that the production limitation is also in our side. It's a very complex vaccine to produce, but we are planning to have 40 million doses of the vaccine, single dose, so 40 million people vaccinated in 2026.
Okay. So unfortunately, we ran out of time for this seminar. So I would like to close the session thanking again our 3 speakers and all of you for attending and your active participation. Thank you all.